Good morning, guys. Good afternoon, guys. Sorry, it's all confusing about this stuff. I think this time will be a bit faster. We do not need those qsub command line to wait for this, but I just review from the new additions to the course website. I add some visualization instruction with IGV. You can just check it out in the course website. That is just by the for already downloaded files. You can just visualize it through these 5 steps, one by one, and then you can visualize it as we got it last week. And then I just quickly go to the section that we finished last week with the count reads that you did and sent actually a qsub through ACF, and you may or might got a text file containing the read counts of a BAM file, which that was only from one BAM file. But for the differential expression analysis for today, we need 16 files, and I've already analyzed them. The results is exactly the rest. You already have one text file, and then I added those 15 files. It's in the directory in the course website. You just need to copy them into your local directory, into your current directory on ACF, and then you download them to your local computer, and we start in R and differential expression analysis. So now just go to the directory, to your course directory. Go to RNA-seq lab 2, the previous section, and then HTC that you just created. And it is here, just copy the files that I mentioned in the course website that started with CPR, loss for Haven course, and then end with second underline sec HTC counts. So you should have the folder in here. Just run ls. You will see all 16 files of the text file that we need for differential gene expression for today. Does anybody have a problem in getting the file? So just go ahead and just transfer them to your local computer. And then in your local computer, in desktop, or whatever directory that you saved it, you must have a folder name HTC count containing 16 text file. And we will import them into the R environment, which, if you have already downloaded it, we will load the required packages with required dependencies that this software is needed. Okay. So first of all, we need to define the website to get the packages. EPP622 is the one that we are going to, is the main package of the software that we are going to use it for differential gene expression. And if you get to the point that you need to update, just say no and hit enter. And we also need other packages called color space and grid. Does anybody have problem installing the softwares? And then after getting the softwares, we need to load them into the R by the command library and the name of the package. And if you get the warning message, just don't care about it, it's not important. Create another one. Oh. This one. Yeah.
What are you typing directly?
Oh, sorry. Wait, yeah, I can make it bigger. The first is on the course website. You have to get the files first, the packages first, and then to load them. You need only 3 packages: color space, grid, and EC2. In RNA62. So we start to getting the files. First of all, we define the directory that those text files are located by setwd. I put the files in desktop, then this is easier. And then we define a file name. Which gets the all text files by the command list of files, and it needs a required argument. You should define the path that your files are located, and the pattern, which is the text file. Then you will define sample name, which you just get the names of each file as a sample name with this command. With the sample name command, you just ask for, from the software, you will ask that get the name of each file, which is DRR016.125.count. You ask to recognize the first dot and then remove the rest and get the name, which is DRR and the rest for each individual file. And then you get the treatment files and the phenotype one. Just copy the comment from here. Put it wrong. And the way to define a vector and the elements inside the vector is getting the information from your CSV file that I talked about it on the first RNA-seq lab. That in here you can define, okay, this one, for example, DRR 016125 is the name of the file name. This is my phenotype, and this is the stress treatment. So instead of mark, I use control. So if you see, for example, in control ABA solid dehydration, and every 4 elements is again repetition. It's based on the reads that we have, which are in the same order from 25, 26, 27, 28 is the first 4, and if you see the stress is Mach, ABA, and solid dehydration is next. If you see the repetition of again Mach, ABA, solid, and dehydration. So it's for the treatment, and the same for the phenotypes based on these Excel sheet. So again, I'll come back to the course website and copy the treatment, the phenotype. And then you combine all of these 4 together, 4 columns together to create a whole table. And this is our table. If you just type if you see there is a sample name, and this is the file name that this sample is related to. This is our phenotype, and this is our treatment. Which, for example, let's say for this one, 125, the data inside that contains the phenotype wild type and contains the information related to control treatment. And this is for the rest of the reads. This is exactly the same for the other 15 reads. I think you have to choose the directory that you choose your HTTP account. Just search where you located it after you got it from ACF. If you can relocate them specifically on to desktop, you can go ahead with the rest of the analysis. So from here, you start the differential gene expression through the EPP622. And the required element is just the EPP6 data set from HTC count. We put the sample table that I just showed to you, the directory that we are doing the analysis, the current directory, and the design is actually the formula for the statistical formula to design that. Okay, what we are doing with this data. We are just analyzing the treatments and the phenotype effect on our reads. And in this case, it's as Dr. Staten said in the last session, when you put the treatments at the end, it means that the impacts will be verified based on the treatments, the impact of the study. At the end, you will get some genes and some raw count, some statistical power like p-adjusted value based on this formula, which defined by the last character, which is in here treatment. It'll take a couple of seconds to the computer. Okay. So when you get to this error, it asks to verify what your control is. So you have to define in the treatments which one is your control. So with this command, you just define that, okay, in the treatment, the word control is my control actually. And then we run the previous command. And it's essential for every experiment that you have this argument actually between, for example, control and the dehydration experiment. You have to put it in this context. Okay, and in here you will check the number, and because the data will create a lot of zeros, you want to filter out those zeros. So we use this command to filter those zeros. So it's before filtering, it's actually from 16 reads, you have more than 33,000 genes. And when you filter it through this command that you define, okay, remove the ones that have lower than remove the counts that have a mean of smaller than 1. And you rerun the previous command, and it says, okay, you have now 25,000 genes. So it removes automatically all the read counts that have a mean of less than 1. And we will be using these 25,000 genes for the rest of our analysis. So in here, we create an object for the D6 software to start the analysis. And here it starts working. I think it takes about 2 to 3 minutes. This one is an required object from DE622 package. You have to do this command, library DE622, first load the package. In the D6 dataset, you say? Sample name. Just copy the link from the course website. Okay, it seems it's done now, and we put this result in a file called this. And if you type res, it gives you this bunch of information that shows individual genes and lots of other factors that I've already put some explanation on each column. But the main important one for us is this one log2 fold change, which shows the expression level, which the negative values shows the downregulation, and the positive one shows the upregulations of each gene when you are comparing that specific gene with the control. For example, the control with dehydration, control with oxalic acid treatment. And the second important column for us is P adjusted, which shows the statistical power that the analysis give it to us. For example, in this case, I think it's about 90% probability, or yeah, I guess. Anyway, this just shows you how the analyses are going. And for having a summary of the results, how many genes are upregulated, how many genes are downregulated, just type this command, and then it says, okay, about near to 8,000 genes are upregulated, and about 7,500, more than 7,500 are downregulated. And still with this information, we can compare or make a contrast between the treatments and control, or the phenotypes, the wild type, with some commands like this one. It just, these are the genes and their log fold changes, plus their p-adjusted value that shows the samples that were gone under abscisic acid treatment versus control. So you can say, for example, this gene 1010 has 0.20 more expression level than the control, and the rest for the next. For example, another one that has, let's say, the -1 is downregulated, this specific gene. And we can also have this contrast for phenotype, which you just need to change the column that contains the phenotype values, the one as wild type, and the other one that is called rows 1-3. And it again gives you another set of data that you can say that gene, for example, the first one, 1010, it has a 0.43 expression level higher than the control, with the p-adjusted value of this amount. For example, this won't be statistical significant, because we usually tend to use the ones that are lower than 0.1 or 0.05 in our analysis. And in here, some interesting plots, like MA plot, that gives you the whole idea of actually just how many genes and the positions of the genes that have been differentially expressed. It's something like this. If you remember the previous sections, course material, it's exactly the red ones are the differentially expressed genes, and this is the log fold change of 0 as our baseline. We are saying the ones that are negative are downregulated, and the ones that are positive are upregulated. Another interesting plot is PCA plot that shows the differences regardless of the X or Y axis, and it shows, okay, which treatment belongs to what treatment, to what phenotype, and you have to have a R-log, regularized logarithm of the analysis first. This little icon in the top of your R window shows that it's still running. Okay, it's done. Now we plug that analysis by plot PCA command. And give you another kind of plot that shows the variation of the, regardless of the variation of the biological sample, which sample belongs to which treatment and which phenotype. So for example, in this case, you can definitely say, okay, these 4 belongs to the control. So it's 4 different treatments, shows that your data is kind of working and it's exactly what you're expecting. Should be something like this. Shows the variation between the data. And the last plot I'm going to show you here explains an individual gene and its regulation under different treatments or under different phenotypes. I'm just going to be a bit faster, so I'm just copy pasting it. By this command, you will get a single gene that has a minimum level of pre-adjusted value. We ask it to put 2 different figures in one frame, and then we ask to get the data set and plot a figure that shows this specific gene 6760 under its regulation under different treatments. And if you run the second command, it just puts 2 different analysis on one frame. So by, for example, this specific gene, 6760, it says, that's good. Let's say in here, this is based on the treatments, and the right one is based on the phenotype. It says this gene is more expressed, either upregulated or downregulated under different treatments rather than the phenotype. So you can say that the phenotype of this this gene under different phenotype is reacting the same, but not under different treatments. And at the end, you want to extract and export the information that you just generated as a CSV file. So you get the files from the analysis and extract them based on their pre-adjusted value. And then you define a cutoff and extract those reads. Those genes based on the cutoff that is in here is 0.05 for us, which is also reasonable in most of the studies. And at the end, you get the raw data, the data that's just being caught and filtered, and save them into the directory that you are now. And And if you go to the directory, for example, the one that I get the data from, I see the one that I just created, this one, and you get all the data set. On the column A, you will see the gene names, log fold change on column C, and p-adjusted value on column G, which if you see, we put a filter of 0.05, so this is the biggest one, and the smallest one is actually 0. And now you can use this data for the rest of your expression analysis, like pathway enrichment analysis, geo-enrichment analysis, or identification of the specific genes that you are looking for. It's upregulated, downregulated, and then to the rest of your analysis based on these exported file. Okay. I just want to go ahead with this RNA-seq 3, and I try to finish it all. I guess. In this section, we need the software Trinity. I asked the ACF guys, I think 3 weeks ago, they installed it, but every week I found that, okay, there is another problem. I asked them again, they reinstalled another software, and at the end I found, okay, it's better just I do it myself, install it for the Bioconda and Anaconda, and then it's not needed to ask them and disturb them a lot. So I think it's a good thing to start with how to install whatever software you like in ACF without asking for permission, asking too much from them, because some of the packages need a lot of dependencies you need. So it's better to start with software installation in Anaconda. So first, go back to your course directory. Okay, here. You just need to first module load Anaconda 3. In this case, we need three different software: SAML, NumPy, and Bosco. The strategy for installing is all the same, and so we start with the SAML first. You need to create a folder and try to name the folders as the name of the package. Then whenever you want to load them, it's easier for you to follow up what the software you've already installed to load. So in this case, we use just SAML. Okay. And then you activate that folder that you just create, source activate SAML, and if you will get a parenthesis at the end of your command line that it shows that folder is activated. And then you have to go to the website of the Anaconda, and in the search section, just type the name of the package you need to download, the SAML, for example. The way that I usually choose the software in here to download is the one that has the higher amount, the higher number of downloads. So just click on this one, and it gives you a link in here. Just copy and paste it. And then the procedure for downloading the software and installing the software will be completed. Is this the process of installing ongoing for you? And I think it may take about 5 to 10 minutes to install the packages that it needs. And this is only once, unless you delete everything and you want to reinstall it, anything. If you see that it's written package already installed, because I tested it and I've already all of them installed, that's the reason. And you will get this message whenever you want to reinstall the software which is already there. And we will do the same things for NumPy package and the Bosco one. And at the same time, it's ongoing on the background. We will come back to install the other two. Just open another window to follow with the working directories for the RNA Class20. Create a directory called RNA Class20. Create two directories inside the lab tree, raw data, and another one for our analysis. I guess. So go to the raw data. And in this case, we will use two raw reads from a bacterial species. I just got them into a current directory, in a directory that I created. Just copy them in the raw data folder that you are now in. In the meantime, just check the other window if it's already completed for the installing packages document. Now we should have two files, faster files. Go back to the analysis. In this case, I created two substance inside the analysis directory. That was because I don't want to have my raw data inside the analysis folder. So I divided them into different folders, and then I created a kind of a shortcut or a substance inside the analysis. And in this case, we need to again, I think, waiting for those Anaconda softwares to be completed. Do you have them completed? Not yet. Okay. Let's go ahead and start it. And this Trinity software is used for assembly. These two raw reads, we use the required packages and software, especially the Trinity, Jellyfish, 4-type, samples, NumPy, to create an assembly for these two raw reads. And I put two different commands. The first one, again, we need to wait, but that is the reason I created the second one. We do not need to wait. We can just submit a job through a .qsh command. So the first one and the second one, the difference is that you need to only waiting. So if there, just go with the second one. If you have already finished those packages installed, we can go ahead. If not, we can wait for a few seconds. Yeah, NumPy and the second one, yes, Roscoe. It doesn't take too long for the rest. The first one is taking long because you haven't already installed the Anaconda in your directory. That's why. So let's just talk about about the arguments, the parameters that is needed by the Trinity software. It asks for the type of the sequence, so it's FASTQ file, the maximum memory, because if you do not define the memory of that device that you are loading your analysis on, it just uses an over uses of the memory. So you have to define a specific memory. Then the left and the right reads. And the SQLite type means the FR, it's FR, RF or FR, which means that, for example, it's a forward and reverse. In our case, it's exactly FR, means that forward read is the number 1, and the second one is the reverse read. And the number of the CPU in this case we define as 1. The mean config length is the 51, means 50 nucleotides. It means that the assembly software, the assembler will remove the reads that have less than 50 nucleotides and give us the ones that are higher than this. And the Jacquard clip, no salmon, and Trimomatic are 3 other options that are used for removing the adapter and using the option for the reads that have overlay of 3' UTR, too many 3' UTR. It will just adjust those amounts. And no salmon is just we do not want to use the software salmon in this case. So these are all the required and optional arguments for the Trinity. And the second code is actually just similar to that one, but just adding some arguments to run the software, to run the job through ACF. And again, you can run it. You can get the data maybe in 2 days, 3 days. But again, I just got the data. I have them. If you have a free window in here, just go ahead and copy the results that they're gonna generated from the course website in the results section of RNA-seq Lab 3 section. After copying it, you will have a file called copy.fasta. Has anybody finished installing those Anaconda software? Did any? Okay. And one point about this Anaconda, like the ACF, when you want to use and load the software, you use module load, let's say Trinity. In this Anaconda, you always use source activate, the name of the package, and then it will automatically be loaded. When you want to check after you get your assembly done with the Trinity, you want to check how many sequences, how many reads that assembler have generated for you. You use the command grep C and the software. And the number of transfer file. It shows you 2,324. That shows the number of the assembled reads from the Trinity. They're just waiting for the. I went through the software Anaconda, installing through Anaconda. For the first time we want to install it, it takes a bit, maybe 10 to 15 minutes. And I think most of the students are waiting for that one. And just short tips about these two separate softwares that are optional software to check how good your quality of your assembly is. We use the transrate and the passcode. Transrate use your assembled read with the raw read and say, okay, 97% of the assembly is available in the raw reads, or maybe less or low. Assign a specific score to your read and say if it is higher, it's between 0 to 1. If it is close to 1, it means that your reads are perfectly assembled, or if it's close to 0, so it means that the assembly hasn't done a good job. And for Botso, it just search the Protobacteria ortholog, which actually the orthologs are the genes with different variation but have the same function. And in this case, the bacteria that we are using for assembly are from Protobacteria section. So we just kind of mapped and aligned them, the genes that are available in those ortholog database with the assembled data set that we have. And then at the end, it says, okay, 70% or 80% of your genes have an ORF, open reading frames, that are similar, or the similar functionality with the ortholog database, of this case, Protobacteria. So you can define that my assembly has a good number of ORF found based on a Protobacteria, let's say, ortholog database. So by this tool, you just say my nucleotide, the assembly from the aspect of nucleotide is good, and also from the aspect of the protein is also good. And that's why we use this tool to kind of verify and confirm the assembly is working well for us. Another point for this source activation through the Anaconda, whenever you activate the software, it's better to deactivate it after you finish off this analysis by that software, because it will just pile up together, and then it will reduce the memory that you want to use for a specific software. So it's a source activate salmon. When your analysis is done, this type source deactivate, and then it's deactivated the software. And for the Trinity, if you want to run the software, you have to define it even multiple software at the same time, which is fine. But be careful that when your analysis is done, just deactivate all of them one by one. Source deactivate Salmon, source deactivate NumPy, source deactivate Trinity. Is it process for you?
copy the change, copy that's the file, it's edit, and see it in your directory that won't undo it.
For the Trinity software, since some of the dependencies are lacking in here, but the original assembler is there. So let's say we are gonna just load the software, and then type Trinity. It gives you a whole idea of what are the required and optional elements. Just type after your module load Trinity with this command, module load Trinity, then type Trinity with capital T. Then hit enter, you will get list of information that is good when you have different data sets, like whatever different lengths you want to change, the number of CPU you want to use, the different max memory or whatever. So in here, let's create the qsub command for the Trinity that we wanted to submit it through ACF. Let's just run this, write this command. This is the current environment that we are working on, and ask for executing based on the current environment, the bash. And then we defined another element as OE, means that give us an output file, whether it's completed, whether there is an error. Just tell us what's the error in a text file. The project number, the course website, the course project. Notes one. In this case, I asked for a wall time of 4 hours and 30 minutes because I tested it. It takes about less than 5 hours, more than 4 hours is needed. That's why the job will be killed. So your analysis is better to have a higher amount of wall time. So you define the space, that the location that your analysis is going to be done by the current working directory, and then the softwares and the dependencies that are needed to be loaded. Which is load jellyfish. Load 3.2. And cruise. And after loading the software and dependencies, we start the software by defining the required and optional element. One of the newly added features to the latest software of Trinity is that they added a Trigonomatic module inside their software, so you do not need to trim the reads first and then do the assembly. You just need to define the Trigonomatic argument and then just supply the reads, and then it automatically identify the usual adapters that are used in the dataset, the raw reads, and then it will clip them out, and then you have your assembly, which the parameters that it's used is this one, which is automatically in the software. And if you want to change it based on the, I don't know, the length of the window, you want to have to clip more nucleotides, to leave more nucleotides, you can go and define it specifically. Then save the file. Make it executable. Did you get it done or still waiting? Somebody? So did you get those first downloaded or not? Salmon and NumPy. Okay. Yes, exactly. That's what I just realized. That if you have the first two, you can go ahead with the Trinity, and then just as I just showed to you, create that qsub command, and then make it executable, and then just send it to the ACF. Write qsub Trinity assembly from the directory that you have the software for the raw read and the your FASTA file actually. It's your command that finish with .qsh. Just send it. Okay. And I don't know if you may be locking, you can get it maybe by tonight, or not locking, you can get it next week. But the result will be the same with the Trinity-copy.factor, exactly the results. And then further to go with the next section, which is it's great. Since Bosco is still ongoing for some of you, so please open another window. Okay, just create a folder inside the RNA-seq lab 3 called translate. Feed into it. And here, activate the software, force activate salmon. You will get the parentheses, says that the salmon is activated. And in here, type this one. I think in the course website, I provided a link in the section of the translate and the bullet that mentioned run the software. There is 2 lines. The third line is Haven courses and blah, blah, blah. You do not care about it, just use the translate. Just do not care about the rest. Just type this one that I'm just writing in here. So the command asks for the assembly. So you have to provide the assembly, the FASTA file that just created by the software. So it's in the here. The second argument asks for the left read. Nope. That's exactly the one that you should follow. Yep, it's need only let's just run to make sure that it solves. Okay. In here, before running this command, module load translate. Just type module load translate, and then hit enter and type translate. You should get the health information. And then start typing the argument. Define the left and right. Another optional one, like whether start. We avoid getting some unnecessary information during the analysis. Are you getting this thing? Oh, it's this one. Lab, yep. RNA-63. So for this translate software, we need the SAML and the software translate. I will update the content of this section. And for the Bosco, I think it takes about 10 minutes or maybe 15 minutes to be completed. For the last part, which is Bosco, the required argument, first you have to download the file from the Bosco website that contains the ortholog database for the Protobacteria, and then the essential arguments which are needed is the FASTA file that you just created by the assembler, and those orthologs which you get from the Bosco website, and you define a type of the assembly that is a genome assembly, and then you define a output folder for the analysis that you will get the data out of it. And since I think we won't have time to get this part done, Oh, it's already done. So just go to the result folder and then check the assembly.csv file. But let's quickly adjust again. It's not a good thing, but just copy paste the file to make it more readable instead of downloading. You can download this assembly.csv into your desktop and visualize this through the Excel. But this one exactly show you the results that we need to interpret. Okay, here it says that out of 2,324 sequences, we have the alignment of okay, shows the mean or percent, the number of the sequences that have the specific length of over 1K, over 10K, and the result, the rest of the metrics that you can find specifically into the Translate website. And I think the number that is useful for us in this case is I don't know where it is. But in summary, this is the shows how good quality your assembly is. And you can find more metrics description. It's not here. You can define it as what you need from your assembly. And since I'm not sure whether you have that file or the Bosco already downloaded or not, but the output of that analysis is something like this. That, for example, in our case, in this Protobacteria ortholog database and our raw read, it says 98.6% of your assembly that you just generated is similar and exactly the ortholog database from this Protobacteria. It shows a good amount of, it's a good quality of the assembly from the aspect of the ORS, the ortholog. And at the end, just source deactivate summon. How many of these are waiting for that possible? So that was the last command I ran and it worked. Wrong basket. Okay. So that's all what I have for today, guys. I think just after getting that downloaded, follow the password, that the command lines, and get get those files done, and also upload the last results that I generated from that file into the course website to make sure that you're getting the exact results out of it. Thank you.